Hello everyone. In this particular class, let us study about prevention of fungal growth. You know, the fungi has got various economical values. Apart from their importance, there are certain harmful effects also, where they are involved in the process of biodeterioration. They are involved in spoilage of food, timber, leather, paper, textiles, and also they are also found to be pathogens onto animals and plants, because of which there would be a great economical loss. Hence, in order to avoid this, the prevention of fungal growth has to be done. It can be prevented by following methods: by the process of sterilization, that is, by the process of killing the fungi using some physical agents, or by providing the conditions, unfavorable condition for the growth using antifungal agents. First, let us study about the sterilization or killing this particular fungi using heat treatment and by ionizing radiation. First, let us study about heat sterilization. There are two different methods where we can use the heat in order to sterilize or to kill the fungi. One is by moist heat method. So in this method, coagulation of protein happen, where the proteins get coagulated, which results in the death of the fungi. The different methods of moist heat sterilization are hot water below boiling point, boiling water, steam sterilization. Hot water below boiling point, there are two different methods, that is pasteurization. You know about pasteurization. There are three different methods in which the pasteurization can be done. It has been elaborated over here, that is at high temperature and short time, 71.7 degrees Celsius for 15 seconds, less temperature and long time, 63 degrees Celsius for 30 minutes, ultra high temperature, that is 135 to 150 degrees Celsius for 2 seconds, and chilling immediately. These are the three different methods of pasteurization. And you have incipation, that is boiling at 80 to 85 degrees Celsius for 3 consecutive days. So for example, so today you are going to boil the thing which has to be sterilized at 80 to 85 degrees Celsius, and you are going to incubate or leave it for 1 day, and again you are going to boil it again. So like that 3 consecutive days we are going to do. Hence, during that particular time, the organism which can withstand the temperature, which cannot withstand that particular higher temperature, they get destroyed. And the others which can, some of the spores which can withstand that particular temperature, they are allowed to incubate, where they germinate, and again you are going to boil at that 80 to 85 degrees Celsius, they also get destroyed. So if you are going to do such things for 3 consecutive days, the sterilization or the removal of the fungi or any of the bacteria can happen. That particular process is called as incipation. And you have got tyndallization. This is also called as fractional sterilization. So here we are going to boil the things. For example, any of some of the liquid media where they have got some protein, where the coagulation should not happen, such kind of or certain antibiotics or serum or any of such some vaccines, if we have to sterilize, so we cannot go for higher temperature. Hence we can go for either fractional sterilization or we can go for such incipation. So as I was mentioning, tyndallization is also called as fractional sterilization. Here we are going to boil at 100 degrees Celsius, that is boiling point of water, for 15 minutes, and incubation period of 3 days in between. 3 days we are going to incubate, and then again you are going to boil. So if there are any dormant spores or something, they germinate, and again you are going to tyndallize or boil, again they get destroyed, and the cycle continues. So that particular process is called as tyndallization. Autoclave, we know, you are going to sterilize uh, the required things in an equipment called as autoclave, where we are going to generate the steam under pressure at where we are going to sterilize at 121 degrees Celsius at 15 pounds per square inch for 15 minutes. So these are the different methods of sterilization, heat sterilization, that is moist heat method of sterilization. We have dry heat method of sterilization. So here the main mechanism is denaturation of protein happen, that is without the involvement of the moisture. Only with the help of heat we are going to destroy the protein. Because of the denaturation of the protein, the lysis of organism happen, and at the same time the high temperature may also burn this particular organism into ashes, as in case of incineration. The different methods of dry heat method of sterilization is red heat, that is we are going to heat the substance which has to be sterilized using the Bunsen burner. The flame of the burner we are going to use, and you are going to heat till it become red hot. That particular method, where at that particular point of time the organisms which are there, they get destroyed. Flaming, we are going to flame is a type of method of sterilization where the metallic objects are being sterilized in this particular method, where the instrument we are going to dip in alcohol and then we are just going to flame it on the gas flame. That is called as flaming. Incineration. So here in this particular case, say for example any of the disposals, hospital disposals, or any of the residue are just going to burn it, or any other waste we are going to burn it into ashes. That particular process is called as incineration. Infrared radiation. Here in this method, infrared radiation, here the thermal, this is also a method of thermal sterilization in which the radiation is absorbed and then converted into heat energy. The infrared radiation is being absorbed by the object, and then that particular radiation is converted into heat energy. So during this moment the instrument will be exposed to radiation, which will result in the temperature of about 180 degrees Celsius for about 17 minutes. At that particular temperature, for that particular period, what happened, the entire microorganisms or the fungal organisms which are present in that particular surface, they get destroyed. And this particular method of sterilization is applicable for packed items as well as syringes, catheters, etc. And hot air oven, this is on one of the common method of sterilization, where we have we'll be having the equipment, hot air ovens, where we are the things, glasswares or whatever has to be sterilized, we are going to keep there and set the temperature to 170 degrees Celsius for 30 minutes, or 160 for 60 minutes, or 150 degrees Celsius for 150 minutes. So likewise here the microorganisms, they get destroyed by the denaturation of protein, and also they get burnt into ashes. This is about dry heat method of sterilization. The dry heat method of sterilization includes red heat, flaming, incineration, infrared radiation, hot air oven. Then coming to the usage of ionizing radiation. So one of the common method of killing the microorganism is by usage of UV radiation. So in this particular method, what happen is if the UV radiation is being exposed onto the organism, we are talking about the fungi, they make a direct hit on the nucleus, where they make a direct hit especially on the DNA, where the UV radiation results in the formation of pyrimidine dimers. Pyrimidine dimer formation happen. Because of the formation of such pyrimidine dimer, this results, or this actually causes the DNA replication is being avoided. So once again I will repeat here, once the UV radiation is being exposed on the microorganism, what happened, this radiation make a direct hit on the nucleus, direct hit on the DNA, results in the formation of pyrimidine dimer. This pyrimidine dimer interferes with the DNA replication. That is, the DNA replication is being stopped, or which results in the process of mutation, because of which the appropriate protein synthesis has been not formed, which results in the mutation as well as the death of organism. Then, provision of the unfavorable condition for the growth of fungi. Many fungi grow poorly or not at all in absence of oxygen. Hence, oxygen deprivation can sometimes be used in the to prevent the fungal growth. It means lowering the oxygen concentration prevents the growth of fungi that cause spoilage. That is, if we are creating, especially in some of the packed food items and all, the oxygen, here what they are going to do is they are sealing that particular container so that the oxygen entry is being stopped. If the oxygen penetration or the oxygen concentration is less in that particular thing, the fungi cannot grow at all. Hence, we are providing such unfavorable condition so that the growth of fungi can be inhibited. Say for example, when we are going to store the grains in this sealed container with little space, so the oxygen level is less, the concentration of oxygen is less. Hence the metabolism of the fungi will also lowers. So we are going to inhibit the growth of fungi by providing the less oxygen or no oxygen in that particular region. And also lower the water activity will prevent the fungal growth. Say for example, if we have to store any grains or any other things, so if there is some moisture, the fungal growth happen. So if you are drying that particular grains or whatever the food crops and keep in a appropriate container, what happens? The fungal growth has been avoided. Low temperature, if you are going to provide the low temperature, especially the refrigeration or the cold rooms or cold storage, whatever it is, if you are going to store in such condition, the fungal growth has been delayed. Hence, if we are going to provide certain unfavorable condition, that is lowering the oxygen concentration or lowering the water activity or lowering the temperature, will inhibit the growth of fungi. Then coming to the antifungal agents. So antifungal agents, these are certain chemicals which we are going to use to control the fungi. They are also called as fungicides. If you are going to use it for plant, it is called as fungicides. These fungicides which we are using, that is antifungal agents, they have got a mode of action. They have each of these antifungal agents differ in their chemical nature and their properties and their mode of action. So let us study the mode of action of some of the fungicides, antifungal agents. So for example, if this is a fungal hyphae, and this is the cell which we have elaborated over here. The target of this antifungal agents can be the cell wall, cell membrane, cytoplasm, nucleus, or any other organelles. So let us take few examples and let us study how actually they may affect. What is the mode of action? Say for example, thiophanate methyl, they affects the nuclear division. So this is one of the fungicide or antifungal agent, that is thiophanate methyl. They affects the nuclear division. Likewise, you have metalax. This also affects the nucleic acid synthesis. This thiophanate affects the nuclear division, whereas metalaxyl, that affects the nucleic acid synthesis. Likewise, you have certain sulfur based fungicides. They affects the mitochondrial function. Remember, they affects the mitochondrial function. Some of the copper based fungicides, they affects the function of the proteins and enzymes. Copper based copper-based fungicides, sulfur-based fungicides, they affect the mitochondria, whereas copper-based fungicides, they affects the protein and the enzyme. Likewise, you have potassium bicarbonates where that affects the plasma membrane, that affects the plasma membrane or the cell membrane of the fungi, whereas there are certain fungicides where they affects the cell wall. Say for example, dimethomorph, they affects the cell wall function. So these are some of the fungicides or antifungal agents where the mode of action may be on the nucleus or on the cell wall or on the cell membrane or a mitochondria or a protein synthesis, etc. So now let us study each one of the antifungal agents in detail. First, let us study about the elemental sulfur. This is one of the first fungicide which has been developed for powdery mildew disease. So here the infected plant, the plant which has been infected with the powdery mildews, they have been dusted with the sulfur formulation. What is the mode of action of this sulfur? Why we are going to use sulfur as an antifungal agent? So as I was mentioning in my previous slide also, this sulfur which we are using, it can accept the electron from the cytochrome. You know, I said the sulfur inhibits the activity of mitochondria. So in my previous slide I was explaining. So how actually it will affect the activity of the mitochondria? You know, mitochondria is the powerhouse of the cell. They are the powerhouse of the cell. So what this there what happen, electron transport system happen where the ATP generation and all happen, that we know. So here what the sulfur does, once the sulfur has been dusted, the sulfur enters into the cell. They enter into the mitochondria where it accepts the electron from the cytochrome. They accept the electron from the cytochrome, interfering the supply of energy available for the fungal cells. And also the sulfur become reduced. One mode of action is they accept the electron from the cytochrome B and interferes with the energy supply of energy to the fungal cell. And also the sulfur become reduced. By accepting these electrons, they become reduced and they get converted into hydrogen sulfide. Hydrogen sulfide is found to be toxic to that of a fungal cell. And apart from that, they produce insoluble sulfides where they combine with the metals, the cofactors of the enzymes which are there. They combine with them and form insoluble sulfides, and hence they affects the physiology of this particular fungal cell, which leads to the death of a fungi. So once again I will repeat, the elemental sulfur is one of a fungicide which has been exclusively used for powdery mildew infections. The mode of action is, since this particular sulfur once it has been applied onto the plant, this particular sulfur enters into the fungal cell, enters into the mitochondria or into the cytoplasm, accepts the electron from the cytochrome, and they become reduced to hydrogen sulfide, and at the same time energy supplied to the fungal cell has been interfered, and also it produces insoluble sulfides. Hence they form a toxic substance inside the fungal cell, which results in the destruction of the fungi. Likewise, we have sulfur dioxide. It is used to prevent the spoilage of wines, fruit juices by yeast and molds. It is being used, it may be added as sulfur dioxide or sulfides or bisulfate. Sodium metabisulfite, these are some of the preservatives which we are going to use in the preparation of fruit juices and all, isn't it? Likewise, we can use this sulfur dioxide or sulfite or bisulfite as a preservative during the preparation of fruit juices, squashes, etc. All these here the main mode of action is the pH alteration. So at that particular pH, the microorganisms, say for example Saccharomyces cerevisiae or Candida, they, because of the alteration of the pH, this leads to the chemical change in the cell component, which leads to the death of organism. And you have Bordeaux mixture. This is one of the fungicide which was formulated by Milladent from Bordeaux University, and this is the composition of this Bordeaux mixture is the copper sulfate and calcium hydroxide and water in the ratio 1:1:100. So that is the formulation which was done by Milladent. So this has got copper in that particular mixture. So what this copper does, this particular Bordeaux mixture prevents or penetrate inside the fungal spore, bind to the amino carboxyl and thio group of protein, and inhibit the enzyme action and prevent the germination of the spores. So Bordeaux mixture is one of the very good fungicide, and it has been it has got copper sulfate and calcium hydroxide in it. The main mode of action of destroying the pest or the fungi is by penetration of this particular substance, this particular Bordeaux mixture inside the fungal spore, and it binds to the amino carboxyl and thio groups of proteins. In my previous slides I had told that copper based fungicides, they alters the properties of the protein. Hence here, this, because this is one of the copper based fungicide, this particular fungicide enters into the fungal spore. Once they enter, they binds to the protein, especially the amino carboxyl and thio group of protein, where because of the alteration of the structure of the protein, the activity of that particular enzyme is being inhibited. Because of the inhibition of this particular enzyme activity, the spore germination is also being avoided. Then we can also use weak acids like sorbic acid, benzoic acid, acetic acid. These are some of the is being used as a preservative for food as well as for domestic products. So here the main principle is increase in the concentration of protons, which leads to the cell death through acidification. So here basically we are the acids, they increases the pH in the cytoplasm, which leads to the death of the cell. So where this particular acids gets diffused through the cell membrane and they get dissociated into the cytoplasm, because of the increase in the proton concentration in the cytoplasm leads to the death of the fungal cell. And other fungicides are based upon the mode of action. Let us study inhibitors of mitosis. There are certain fungicides, say for example griseofulvin. This is one of a fungicide which has been produced industrially by using the fungi called as Penicillium griseofulvum and Penicillium patulum. These are the species of Penicillium which is being basically used in the production of griseofulvin. This griseofulvin, the mode of action of this griseofulvin is they inhibit the cell division. So what they does, this griseofulvin will inhibit the cell division. How actually it will inhibit the fungal cell division? They act by preventing the tubulin polymerization and microtubule elongation in the nucleus. So during the process of nuclear division, what should happen? The microtubules should form, and the division of the cytochromes has to go towards the different poles and all. So they inhibit the formation of this tubulin protein. If the tubulin protein itself has been not formed, microtubule formation has been affected. Because of the microtubule is being not formed, so what happened? The cell division, fungal cell division is being inhibited. So this is done by griseofulvin. So hence this, because of this particular property, this griseofulvin is being used in the treatment of certain dermatophytes such as trichophyton, microsporum species. We can use this particular griseofulvin to destroy such kind of infection. So the main mode of action of this cryptofulvin is they inhibit the production of this microtubule formation. And inhibitor of nucleic acid synthesis. There are certain fungicides or antifungal agents, say for example 5-fluorocytosine. This 5-fluorocytosine is being used in the treatment of Candida and Cryptococcus. You know, these are again these are unicellular fungi which causes various infection in case of human. So what is the mode of action of this 5-fluorocytosine? This 5-fluorocytosine is being transported into the fungal cell by cytosine permease. Say for example, if this is a fungal cell, this 5-fluorocytosine, if we are applying on the infected part, this 5-fluorocytosine enters into the fungal cell with the help of an enzyme cytosine permease. With the involvement of this enzyme, they enter into the cytoplasm. Once they enter into the cytoplasm, what happens? This 5-fluorocytosine gets converted into 5-fluorouracil. This 5-fluorouracil which has been formed, they interprets or they interferes in RNA translation and DNA synthesis, because of which the DNA synthesis as well as RNA translation has been interfered, the protein synthesis as well as the DNA synthesis has been avoided, which results in the death of the fungi. So once again I will repeat, 5-fluorocytosine is one of a synthetic drug which has been used against Candida and Cryptococcus. This 5-fluorocytosine, once it come in contact with the fungal cell, they enter into the cytoplasm with the involvement of an enzyme called as cytosine permease. Once they enter into the cytoplasm, they get converted into 5-fluorouracil. With the involvement of an enzyme deaminase, this 5-fluorouracil, it interferes with RNA translation as well as DNA synthesis. There are certain fungicides which are inhibitor of cell wall synthesis chitin. You know, the fungal cell wall is basically made of chitins. If the chitin synthesis itself is been inhibited, there will be a lysis. The cell become very weak and it results in the rupture or the lysis of the cell. So which are those antifungal agents which inhibits the fungal cell wall synthesis? There are certain antifungal agents, say for example poloxin and nicomycin. These are certain antifungal agents which inhibit the chitin synthesis. This particular antifungal agents are produced by actinomycetes called as Streptomyces tendae. This is an organism which has an ability of producing nicomycin. What this particular nicomycin does, they inhibit the production of beta-1,3 glucan synthase. This is an enzyme which is involved. This is very much important in the synthesis of the chitin. If the chitin synthesis has been not happening, what happens to the fungal cell? Which results in the bursting of the fungal hyphal tip. So for example, the growth of the fungi, as we all know, is happening at the tip of the hyphae, where the deposition of the cell wall content happen. If because of the lack of an enzyme called as beta-1,3 glucan synthase, the synthesis of the chitin is been inhibited, because of which the deposition of the chitin onto the cell wall may not happen, which results in weakening of the cell wall of the fungi, resulting in the bursting of the hyphal tip. Because of the bursting of the hyphal tip, what happens? The fungi growth is being avoided, stop, and finally the death of the fungi happen. And also there are certain fungicides which inhibits the protein synthesis. So for example, there are certain antifungal agents, or say for example casugamycin. This is one of the antifungal agent which has been extensively used against the rice blast disease, which is being caused by Pyracularia oryzae. This is one of fungi which cause blast disease in case of rice. This particular Kasugamycin is being isolated from Streptomyces kasugaensis. So what this particular Kasugamycin does? So here, in this particular image, you could see the normal, the protein synthesis, how it happened. So during once the mRNA come and bind onto the ribosome, um, during the process of translation, RNA bring the amino acids and uh, it will bind onto the mRNA, and the process continues. So here what happened, this Kasugamycin does is, it interferes, it prevents the binding of this amino acid tRNA onto the ribosome. So what happened here is, this tRNA, for example, this is the tRNA, amino acid tRNA. This is supposed to come and bind onto the ribosome. That is, it has to bind and leave the amino acid over there. So this Kasugamycin will gets binded itself onto the ribosome. So it blocks the site where actually the tRNA was supposed to come and bind. Hence, the blockage of the initiation of protein synthesis is being blocked. Hence, this affects the protein synthesis. So, this Kasugamycin, how actually it inhibit the growth of Pyracularia oryzae is, this particular antifungal agent bind onto the ribosome, blocking the site of the tRNA, amino acid tRNA, which was supposed to bind and start with the initiation of protein synthesis. So this is about the mode of action of Kasugamycin. You have Nystatin. This is one of a very good antifungal agent. Nystatin was first isolated from Streptomyces norsci by Elizabeth Lee Hazen and Rachel Fuller Brown. They are the one to isolate this particular antifungal agent from Streptomyces norsci. So the main mode of action of this Nystatin is, this Nystatin binds to the ergosterol. So what is this ergosterol? This ergosterol is the main component of a fungal cell membrane. You know that. Say for example, if this is a fungal cell membrane, not the cell wall, cell membrane, they have got a sterol in them. They are the ergosterol. Apart from this phospholipids, we have some sterols called as ergosterol. That is a distinguishing characteristic feature of the fungi also, that is having the ergosterol in their cell membrane. So what this particular Nystatin does, it binds onto this particular ergosterol, because because it binds onto this particular ergosterol, and it binds and releases this ergosterol, because of which what happened, the pores are created on the cell membrane. Because of the creation of certain pore, what happened, it is easy for the leakage of the cytoplasmic content. That is, the cytoplasmic content is being leaked out from the fungal cell. So for example, in this particular image, you could see the cell membrane having the phospholipid membrane. It is a phospholipid membrane, and there are certain ergosterol also along with this phospholipids. So this, once we are going to treat this with Nystatin, this is one of a polyene substance. This interacts with the ergosterol which is present on the cell membrane, and removes that particular ergosterol and creates the pore. See, the pore has been created. Once the pore has been created, the potassium and other small molecules are lost through this pore, causing the cell death. The cytoplasmic content, especially the potassium and other important small molecules, are leaked out from the cell of the fungal cell, which results in the death of the cell. That is the mode of action of nystatin. And this particular nystatin is one of a drug which has been extensively used, especially for candidal infections in intestine. And nystatin is also available as topical creams and can be used for superficial candidal infection on the skin also. So likewise, you have one more important antifungal agent called as amphotericin B. This is now a talk all over at this particular outbreak of this corona. After the infection with the corona, there are certain black molds, especially this mucor or the zygomycetes fungi, which are infecting those people where especially they are immunocompromised patients, as well as in some of the diabetic patients, that there is one more outbreak of after this corona. After their treatment, they are using certain sterols, because of which there are the growth of sterols. They are because of which what is happening, the chances of getting infection is high. The one of the treatment for this is the usage of antifungal drug called as amphotericin B. This is also one of the antifungal agent which is being used in serious fungal infection, as well as in some reports tell that it is also being used in leishmaniasis. The fungal infections, for example, aspergillosis, blastomycosis, candidiasis, coccidiomycosis, mucormycosis, cryptomycosis, zygomycosis, in such infection, this particular antifungal agent has been used. This amphotericin B has been isolated from Streptomyces nodosus. Remember, Streptomyces nodosus in 1955, and it has been used for medicine from 1958, and even WHO has also listed out it as an essential medicine, and it is also one of the safe and most effective medicine in our health system. So how actually what is the mode of action of this amphotericin B? This amphotericin B is being found as a lipid formulation. It is found as a lipid formulation. Amphotericin B, once it comes in contact with that of a cell wall and the cell membrane, they enters to the cell membrane and it targets the the ergosterol. As I was mentioning before also, this is one of the cell membrane is made up of the apart from phospholipids, there are some sterols also, that is ergosterol is present in the cell membrane of this fungal. They get complexed with this ergosterol, and this ergosterol has been removed from the cell membrane, because of which the pores have been created. Because of the creation of the pores, what happened, the protons as well as the essential molecules, important small molecules, are leaked out from this particular cell membrane, which alters the permeability of this fungal cells because of the leakage of the cell wall component, which results in the death or of this particular fungi. This is the mode of action of amphotericin B. So this is very important drug nowadays. It has been which has been used against mucormycosis or zygomycosis, which is one of the the outbreak all over. So the mode of action is it's bind to the fungal ergosterol, which is one of the primary sterol in the fungal cell membrane, and because of which it creates the pore, which because of the pore creation, the permeability of the membrane is being altered, which results in the leakage of the cytoplasm, which results in the death of the fungi. So this is about amphotericin. So in this particular video lecture, we studied the different methods of the preservation of the fungal growth, starting from the sterilization, usage of moist heat method of sterilization, dry heat method of sterilization, usage of UV radiation, and finally we studied the different fungicides, antifungal agents and their mode of action, and how actually they play an important role in destroying or the prevention of this fungal growth. Thank you.